Hi everyone, welcome to my YouTube channel. In this video, we are going to be talking about DNA replication. I'll make another video for transcription and translation later on, but let's just focus on replication for today. So in order to first understand why this whole process is occurring, we need to understand the cell cycle. So the cell cycle has multiple phases. We have the G1 phase, we have the S phase, we have the G2 phase, and we have the M phase. So G1 and G2 are checkpoint phases, but I like to think of it that the G stands for growth. So yes, there's checkpoints happening here, but it's also mainly for growth. And the S is going to be for synthesis of DNA. So AKA that's going to just be DNA replication. So what we're talking about today is going to be what's occurring in the S phase. Excuse my background noise. G1 is going to be for growth, and in the first growth phase, we are going to just be creating all the proteins, carbs, lipids that are needed for replication. We're just getting prepared. S is the actual replication process. Next up, we have G2, which is the second growth phase. In this phase, now we're just getting ready for cell division, and the chromosomes are going to start to be visible. DNA is going to tightly coil into them. Tightly coiled DNA, and then the M is for mitosis. Mitosis is PMAT, which is cell division. I have a separate video on mitosis and meiosis, and you can check that out later if you need to. Okay, so now that we know this, let's just quickly cover the central dogma theory. So the central dogma basically says that we go from DNA to RNA to proteins. And as we know, these proteins and the way that they're folded and their different functions are how we are able to function as human beings, and how different parts of our body are able to perform different functions that they need to perform. So in the central dogma, DNA to RNA, that is the process of transcription, and RNA to proteins is the process of translation. And as I said, I'll make another video in the future about transcription and translation. For today, we are going to focus on DNA replication. So I'm just going to make my own addendum in which we're just going from DNA to DNA. We're going to replicate it, and then we're going to transcribe it into RNA, and then we're going to translate it into proteins, and these proteins are going to go and do their functions. Something to note is that while we are replicating DNA, while we are dividing cells, the size of our genome never changes. Genome never changes. So if you are going to be asked an exam question that is going to have answer choices pertaining to, oh, genome doubled in size, genome tripled in size, genome has been cut in half, if you see an answer choice like that, immediately cross it off because the size of our genome does not change. Okay, so now let's talk about DNA replication. So as you know, DNA is in the form of a double helix, and on these sides we have phosphodiester linkages which create the phosphate backbone of the DNA. This phosphate backbone is on the outside. So would you expect it to be hydrophilic or hydrophobic? If you said hydrophilic, you are right. Phosphodiester backbone is going to be hydrophilic because we have all of those phosphates, and those phosphates are negatively charged. Phosphate backbone is negatively charged. And connecting these phosphate backbones, we have our base pairs that are connected together. So those are our A, T, G, C, and A will pair with T, T will pair with A, G pairs with C, C pairs with G. Those are what are making these bonds over here. So we could have like an A over there, a T over there, G over there, C over there, like that. And the bonds in between these bases, these are going to be hydrogen bonds. Something to note is that A and T only has one hydrogen bond, whereas G and C have two hydrogen bonds. So that's why in strands that have a higher composition of G and C, they're harder to break apart or denature. It's harder to denature that DNA. Whereas if it has more A and T, it's a little easier to mess with that DNA because it's not as strong as the G and C bonds. These are hydrogen bonds. If you can see, these are all on the inside of the DNA. So would you expect this to be hydrophilic or hydrophobic? If you said hydrophobic, you are correct. I just like to think of it in the same way that we think of the phospholipid bilayer. The heads, which are pointing to the outside, those are hydrophilic, but then you have the tails that are pointing towards the inside, away from the outside space, and those are the hydrophobic tails. Same thing over here. Okay, so if we want to do our DNA replication, then we can't have all of this torsional strain. We can't have it all tightly looped up because then we really can't get in there and replicate. So we are going to have something called topoisomerase, which is basically going to make little nicks in DNA in order to relieve this torsional strain, in order to make it open up like this. We're going to make it into, that's not a very good depiction. Basically, so if we have it like this, we're going to make it like this. We're going to make some nicks, and then we're going to glue it back together, and now it's not all spun and twisted and knotty anymore. Now it has relief of torsional strain. So once we do that, now you can get to this picture, which I'm sure you all have seen before. And the way that we are kind of unzipping the DNA and making this top template leading strand and the bottom lagging strand kind of separate and visible to each other, we are using an enzyme called DNA helicase. So I just like to think of topoisomerase. T stands for torsion. DNA helicase, I just think of this like a zipper. So it is basically unzipping the DNA and is going to expose all of these base pairs. So as the DNA has unzipped, we're going to have this molecule called, or this enzyme, called DNA polymerase, which is going to start adding the base pairs. But in order to add those base pairs, we need to kind of understand the directionality of the DNA. So as you know, DNA is always translated and transcribed and read 5' to 3'. And both strands of the DNA are anti-parallel. So if the top strand, our leading strand, is 5' to 3', then the bottom must be 3', 5'. But when it gets read, it's still going to go 5' to 3', always. This is going to be the lagging strand. And we have our base pairs. So when we start to build a new strand, it is going to go like in this direction for the leading strand and in this direction for the lagging strand. Now you might be wondering, I drew the arrow from 3' to 5'. So how is that following the 5' to 3' rule? Well, the rule is in which you are adding the new polymerase, or the new base pairs. In which case, it's anti-parallel again. So now 3' is going to pair with 5', 5' is going to pair with 3'. So it's being read 5' to 3' in the direction of the new strand. So that's why it's going to be 5' to 3'. And down here it's the opposite. It's 3' on this end, 5' on this end, so it's being read in this direction. And the new base pairs are being added in that direction. DNA polymerase is going to add those base pairs. But can DNA polymerase just get in there and start adding base pairs? No, because DNA polymerase needs to have a primer in order to initiate the translation. And the primer is going to be created by something called DNA primase. And this primer is going to be an RNA primer. So instead of T's, it's going to have U's in it. It's going to be a small segment. It's going to be a small segment of the RNA primer. So then DNA polymerase can start to continue from there. It uses the primer. And then once the use of the primer is done, or we don't need it anymore, a specific DNA polymerase 1, is going to remove the RNA primer. Okay, and as we keep going, and we're going to go in this direction in the lagging strand, we're going to go in this direction in the leading strand, helicase is going to continue unzipping this. So if you can just imagine this continuing to open up, and then it'll open up again more and more and more. This whole thing is going to be called the replication bubble. And if you remember what I said before, way up here, about the hydrogen bonds in between the base pairs, our replication bubble is usually going to initiate where we have more A's and T's, because it's just easier to unzip and unwind closer to the AT region, while GC-rich areas would be harder to start replication just because they're more tightly bonded. So that's just a side note that you should understand. So we have our replication bubble, and we're going to continue going like this. But do you see where there could possibly be an issue? Leading strand, fine. It's going from the 5' to 3' directions. DNA polymerase is going to keep adding those bases, and DNA helicase is going to continue opening up, and then we're just going to continue adding our bases. But what's the issue over here, down at the lagging strand? DNA polymerase is going from 5' to 3'. It's in this direction. Well, we reach the end. So how do we kind of jump back over here in order to make up for what we're missing that the leading strand is getting when the helicase is opening up? We're going to do this by making little fragments. So we're going to go from 5' to 3', make a fragment, come all the way back here, make a fragment, come all the way back here, make a fragment, as helicase is continuing to unzip the DNA. And those fragments are called Okazaki fragments. They're called Okazaki fragments. So these fragments are going to be made, but we need to get them to stick back to each other. Otherwise, we'll just have a bunch of fragments of DNA lying around, which is not very useful for us. We are going to glue them back together with something called DNA ligase. And the way I remember this is that the G in ligase stands for glue. So DNA ligase is going to glue those Okazaki fragments together. And we're just going to keep going. Polymerase is going to kind of jump and pair, jump and pair. It's just going to keep going until we finish our replication. That's not a problem for the leading strand because of the direction. So because there's no Okazaki fragments in the leading strand, everything's going kind of normally the way we would expect it to. It's going to be called continuous replication. Whereas on the bottom, we're kind of like fragment that we have to then glue back together. It's called discontinuous replication. So those are the main basics of DNA replication. I hope that was helpful to you. Definitely I would recommend going to YouTube and finding an animation. Then you can more clearly see what I was telling you about over here with the helicase is unzipping, but on the lagging strand we have to kind of jump back in order to cover what we missed that the leading strand was able to handle. So I recommend finding a good animation video for that. But thank you so much for watching, and let me know if you have any questions in the comments.